you should now be able to see my slides, hopefully.
Yes.
Perfect. So, thanks, Stefan, for the very nice introduction into volume rendering. So I'll pick up from here. And while most of the examples that, or I think all of the examples that Stefan showed were from medical imaging, medical volume rendering, we'll now move on to the bio part of this. What do we mean by that? Well, you might have seen images like this, molecular graphics, and that's actually has been for quite some years a large part of my research. So I will talk about volume visualization for molecular graphics and how this volume visualization techniques can be used to analyze, explore molecular data. And of course, one source of data is similar to the medical domain is imaging data. So you can actually, so to say, shoot images, shoot photos or 3D volumes of your molecules using techniques like X-ray crystallography, illustrated here. So you crystallize your biomolecules. Then you measure a certain diffraction pattern. You can reconstruct a density volume map from that, or cryo-EM. I don't want to go into detail about this acquisition method. You basically freeze your protein samples, and then using an electron beam, you can also get imaging data of actual molecules, almost down to the almost down to the atomic level nowadays. So here are two example renderings. Here is that the electron density surface of the electron density map of the small molecule here. Here is the cryo-EM picture, and you can see how the resolution improved between 2013 and today. So as said, almost atomic resolution. However, this is not what I really want to talk about today, because in the end, from a visualization or rendering point of view, the differences to what Stefan just told us is not really there. I mean, it's a 3D volume. You have density values, and you can just render them using direct volume rendering, or if you want to, also maximum intensity projection techniques like that. Other sources of volumetric data in molecular biology could be something like electrostatic potentials. So you have your protein, your charged molecules, here in these two example images, and from these charges you can compute the electrostatic density field. You can show these electrostatic clouds here, or since it's actually a vector field, so at each position in space you encode not just a density value but also a direction. You could also render something like this field lines here. However, again, it is just volume data, so to say. So we are also using the same methods than Stefan just showed us for medical data. So what we're now going to look at in the next 15 or 20 minutes is how we come from a representation like this, what you probably have seen already in some images. So actual atomic data to something like this, a volumetric representation of this data. So here we don't start with something volumetrics, but really discrete positions of atoms in space. And as you know, atoms are usually depicted using this hard sphere model. So each sphere represents one atom here. And this is basically what would molecular simulation give you. So you can have a time-dependent data set of this, so you can see how the atoms move. So the question is, why would we want to use volume rendering, volume visualization methods for that? And how can we use it to create such nice molecular surfaces? And before we look into that, we need one more definition. So as said, here we have our atomic positions here, and we can also call this the van der Waals surface, because this surface of the sphere actually shows the van der Waals radius of each atom, so specific force fields, so to say. And from this, we can infer other molecular surfaces. So here, the solvent excluded surface, ligand excluded surface, and this Gaussian surface. They look very similar, but they need to be computed very differently. I'll come to that in a minute. The only thing that I want to show with this is that these molecular surfaces show important properties of a molecule, because it shows the interface between the molecule and its surroundings. So usually you're interested in, for example, whether small other molecules called ligands can reach some part of the surface and, for example, trigger a reaction. And here, when we use a sphere model, we see all the atom spheres, but we cannot really, it's very hard to judge, okay, can our red ligand really reach a part here? And if we draw such a surface, that's much easier. And we can actually use volume visualization methods to create these three types of surfaces. So again, some definitions here, like I just said, the van der Waals surface just consists of spheres. You have the van der Waals radius of each element, of each atom. You don't consider any ligand or solid molecule, and you get a representation like that. Now, more useful is the so-called solvent excluded surface, which you can see here, which shows the boundary with respect to a certain solvent, or well, at least with respect to a sphere of a certain size. So you take the van der Waals surface, you roll this spherical probe all over the surface, and the probe surface actually traces out the red solvent excluded surface. So everything that is reachable by the sphere surface defines this red, or in this case here, the gray solvent excluded surface. So everything that's beneath the surface, underneath the surface, is not reachable by our ligand that has this radius. Now, as you can see here, you can compute this implicitly. So there are actually three types of geometric primitives, little spherical and toroidal patches that construct the surface. However, that's not what we are going to look at closer, because we want to use volume visualization for that. And by the way, computing this explicit surface patches is quite time consuming. So we are looking for a fast volumetric calculation method to get this smooth solvent excluded surface. And the basic idea here is, again, we use a volumetric grid here to compute a distance field of the solvent excluded surface. So we compute a volume, and at each location of this volume, we have a number, then a value that signifies whether our position is outside of our surface, then we have a positive value, or whether it's underneath, inside the surface, then we have a negative value. So the first step, when we start with the input, are our atomic positions, so just XYZ positions, usually in Cartesian coordinates. And now we, first of all, we discretize our space into a 3D grid. Usually we use a uniform 3D grid, so we have a volume, and our the cells of our volume are little cubes with equal side length. Now, to compute this distance field here, from which we can then extract our surface, we will look at each point of our volume, each point in space, so to say, on our grid, and we classify it. So here you can see the first step of the algorithm. We take the radius here of our probe, of our spherical probe that we want to test. First of all, we test, okay, is the center of our probe actually inside one of the atoms, one of the gray atoms of our protein here? Or is it outside but intersecting? Or is it outside and not intersecting at all? So in the blue part, we are just not touching the protein, the gray protein atoms at all. In the yellow part, we are intersecting them. In the red part, we are inside. So all parts that are inside, you can say, okay, we have negative values here. This would be this part here. In the blue case, we are outside, so we say, okay, we have more than our maximum probe radius. You can just say, okay, that's a large positive value, so to say. But for the in-between case, for the yellow voxels, as we say, for the yellow points in our volume, we need to compute distances here. And we can just do that by, for each of these yellow points here, we search for the closest of the blue points that you know is outside. And then we compute based on distance to the next outside point, the next blue point, we can actually compute this distance field, so the distance of our point to the actual surface, the solvent excluded surface that we want to compute. So again, after doing this, we have a density field that just has negative values inside our solvent excluded surface, and here outside has the distance to the closest atom of the protein. And here beyond this threshold, we just have large positive values. And then we do something we call isosurface rendering. This is very related to what Stefan just told. We can also shoot a ray through our volume, but instead of just accumulating density values, we search for one specific density value. In this case, the point where we cross over, so to say, the distance here to into the density field, in this distance field, sorry, and then we just draw a surface here. Now, of course, here the resolution of the volume, so how fine-grained we sample our volume, is very important. But as you can imagine, the more computations we do, so the more fine-grained our volume is, the slower our computation will get. And here, so you can see the results here. So here we have a coarser grid resolution, 128 cubic voxels. So a grid that's divided into 128 by 128 by 128 little grid cells. And here we just lose some detail. Here, if we double the resolution, we can see, okay, there are actually holes inside our molecule that we just did not see in the coarser resolution. And here, one solution for this could be progressive refinement, so similar than loading some of these JPEGs, where you first get a very coarse resolution, and then it updates to a finer and finer resolution until you get the full image quality. You can also do that here. So you start with a very coarse, very small and fast to compute resolution for your 3D volume. You can render the surface already, and then you can refine it, and after some time you have the full resolution and yeah, all the details that you want to see. So this gives us this very, very nice, very smooth solvent excluded surface renderings, and you can see here. So you can see for this two examples the interface between our protein and some molecule that might attach to it. So here, for example, you can see, okay, this protein here has some cavity where other molecules could go in and trigger some reaction. And this here is an image of a viral capsid. So you could imagine that some drugs that destroy the virus could attach here at the surface or something like this. But again, the solvent excluded surface is just based on a sphere. So it's not really the exact shape of a ligand that might attach here, it's just a sphere that approximates this. So the ligand excluded surface, the second type of molecular surface that I want to talk briefly about here, actually takes the shape of this ligand into account. So we use an actual ligand instead of just one sphere. The computation, again, is very similar. You do this in two steps. So the first step, you just signify, okay, by just using the circumsphere of your ligand, like in the solvent excluded surface, you look, okay, which voxels here in my grid, which grid cells are clearly outside, and here the red ones we have to test further. This could be inside. And then the second step, instead of computing this density field with a sphere, you just sample the exact ligand here. Now you can imagine, of course, the ligand could go in like this, but of course it could also turn around. And for example, here it goes on like this. And to make this computation, we actually pre-compute a table of ligand orientations. So we can, of course, not compute this in good time span for all possible rotations. So we just coarsely sample like 100 different rotations, and then we test against these. And we actually pre-compute here also some kind of density field that we test again. And then we finally get this ligand excluded surface where we really test, okay, could the actual ligand go into this cavity or could touch the surface here in some sort of orientation. Here you can see a comparison. In yellow is the ligand excluded surface, this more exact generalization, so to say, of the solvent excluded surface where we use the actual shape of our red ligand molecule here. Here we have the blue solvent excluded surface of the same molecule. We just have this sphere probe that rolls over the surface, defines the surface. And you can see that there are differences. So the yellow surface gives you a better impression. Okay, could a ligand really reach into here, a ligand of this size? Or is this little cavity actually not accessible? So it is a bit more interesting to a bit more expensive to compute because you have to test against these different orientations of the same ligand. But it's more interesting in a way to the users because they get a better impression. Is there really a reaction possible or not? But as I said, the computation is both very similar. You do this on a volumetric representation. So you use the volume and then you compute this distance field, and then you can just use volume rendering methods to show this. I see we're already running out of time, but I started a little bit later, so I will take 5 extra minutes here. I take that off your break. I hope that's okay for you, because I also quickly want to show the third and last type of surface that I want to show here. These are Gaussian surfaces. And you might know them under the name of metaballs or just blobby or convolution surfaces. It is a more generally applicable implicit surface for particle data sets. But interestingly, in molecular graphics, it can be used to model electron density. So in contrast to this solvent excluded and ligand excluded surface, you don't assume, okay, each atom is a hard sphere, which is of course not physically speaking, but you say, okay, it's just it just models the electron density field around each atom center, so to say. And then we can also compute the isosurface from this electron density field and also extract the surface. And the surface can model this solvent excluded surface closely, but it's not as exact, so it doesn't use this rolling sphere. The idea here of Gaussian surfaces is that you have, as I said, this radial symmetric density function at the center of each atom. So you have the decreasing electron density the further you're away from the atom center. And you define this density field then by just summing up the density contributions of this radial symmetric density kernel from all the atoms in your space. This is the equation. I don't want to go into details here. So it's just a sum of exponential functions here to model this discrete decreasing, this radial symmetric decreasing function here. And here you can see some results. So you have this two, if you then extract the isosurface for a certain iso value here, you can get these images. So you just, if the two spheres are farther away, they're not connected, and the closer they get, the density contributions start to merge here, and then you get this smooth surface that covers both of them. Now, how do we compute this? In theory, of course, you might know that Gaussian density kernels are infinitely wide, but after a certain cutoff radius, the influence will be close to 0. So to speed this computation up, we can just define a cutoff radius, and we don't only have to take particles or atoms into consideration that are within this cutoff radius. So if you look at the density contribution of this blue atom here, beyond this blue cutoff radius, it is close to 0, so we can just neglect it if we sample the density at here or at this point, and just inside this cutoff radius, we have to sample the density, add the density of this blue atom. I also prepared some slide about fast nearest neighbor search. In the interest of time, I will skip a little bit over that. So just to get the rough idea, so what we have to do is to search for nearest neighbors here. So for each point in space here, we have to search for all the neighboring atoms that are within this radius here. This is also not just important for surface calculation, but also for many other applications like clustering also. And we use a grid-based nearest neighbor search for that. As that, well, skip that. But if you're interested in how this works, this is actually what NVIDIA does in their particles demo. So if you look at the CUDA SDK examples and search for the particles demo, this fast GPU-based parallel neighborhood search is explained there. So in the end, what it tells us then, what we can use it for is if we have a point in space, we can just use this fixed radius here, very quickly find all the atoms, all the particles that are close to this, within this cutoff radius, and then we can use this to compute our density volume. So after we have this data structure for fast neighborhood finding, we compute this continuous density field here. So we again do this, of course, on a 3D grid. So here for each grid cell, for each voxel here in our volume, we just compute the neighborhood, we collect the density contributions according to this sum equation that I showed before, and then we can extract an isosurface here for a certain iso value. So if we say this is the isosurface, the iso value for which you want to extract the surface, then we would just get something like this here. So this metaball surface. And this can be parallelized very well. So for each voxel, you can compute this density calculation in parallel, and they can, for example, then again use direct volume rendering to get very nice molecular surfaces like this. And it's actually a very, very fast computation method. So even though it sounds very involved computing all this density volumes, it is actually quite fast. And it's very suitable for visualizing large viral capsids like this HIV capsid that made it to the cover of Nature magazine in 2013 already. There are a couple of applications for that. Again, I don't want to go into details, so you can segment this density volume or the resulting isosurface actually to get some tunnels or pockets in this. So it's not just for showing the data, it's also used for analyzing the data. Or here you can see another example how that you can aggregate the density of some ions here around this DNA, see where they cluster and so on. So I think it was speaking even faster than Stefan here, but I hope I was able to give you a little bit of an impression how this volume rendering, which is of course very prevalent in medical visualization, can also be very useful for molecular visualization, not just to show electron density fields or electrostatic fields, which are density volumes in itself, but also to use them as a means to compute different molecular surfaces, and to use them for visual analysis applications such as this protein cavity detection or the visualization of ion motion and probability density. With this, I'm at the end of my talk. Sorry for almost stealing some of your coffee break, and I'm of course open to questions. So thank you.
Thank you, Michael, for your talk. It was very interesting. I think we don't have time for any questions because the next talk you kind of already took all of the break. Now, there are questions in Discord. Probably you can answer them.
Sure, I'll just look at
Sorry, we have a 15-minute break, so there's some time.
We still have 15 minutes?
That's
okay,
what I thought, yeah. We should be right in the coffee break, right?
Yes.
So if someone wants to stay here in the coffee break and talk to me, otherwise I will look at Discord.
I see one question at least. Maybe we can take that?
Sure.
Do you want to ask it?
Oh, sure.
You just have it.
Yeah. Yichilua asks, During the last computation, should we consider the situation where the ligand enters a cavity and then rotates to go further?
Yes, it's a very, very good question. Let me just quickly go back to the slide to illustrate this. Yeah, the question was if this cavity is deeper, the ligand could, of course, slide in that way and then maybe move inside. With the computation method that I showed here, this is actually not considered. So for each voxel location, we are just testing, okay, is there any orientation in which our ligand could fit in here? We're not looking at a path, but of course you could extend this. This would again, yeah, make the computation a little bit more expensive. But yes, after sampling all these possible orientations, you could then, of course, do a third step, so to say, and see, okay, if I'm at this location, can all the neighboring locations, all the neighboring locations valid for this one? So in theory, yes, you could do this. In practice, no, it's not done in the technique like I explained it.
Okay, I don't see any more questions at the moment. Probably there will pop up more. Thanks again, Michael. Awesome.
Okay, yeah. Thank you, and thanks for having me. And as said, I will look at the Discord. So if any more questions pop up, I can answer them there. So see you. Thanks.
Okay. So we have our break now, and after the break, Oscar will take over. And so I also say goodbye.